Hello, everyone. We are happy to have you here in our advanced webinar on spectral flow cytometry. I'm Nitan, I'm VP New Technologies and Implementation here at MOG. Next to me is Aviva. She's a flow cytometry service team leader, application and scientific support, and she is definitely a flow expert. But before we will dive into our talk today, we just want to give you a teaser on how you can create a really nice and informative analysis of phenotyping panel using Cytec technology. You can see here a full map of 40 markers expressed across all populations. This is truly a milestone for the flow world, as it is the first time that a flow cytometer has been able to detect so many fluorochromes in the same tube. And it makes you curious as to what the future will bring. First, let's go over the agenda for today's talk. We are going to discuss the differences between conventional and spectral flow cytometry. We will show you the spectral signature, how it is created, and what does it tell us. We will explain spectral unmixing and compare it to conventional compensation. We'll discuss the use of autofluorescence extraction and its advantages in dim population analysis. The last part of our talk today, we want to show you the advantages and possibilities that are available with the Aurora 5 laser system. And last but not least, we'll show you the new and updated OMIP 69 with 40 color immune panel that was published recently in Cytometry 2020 Part A. So let's start with a diagram of conventional flow cytometry. We have the sheath fluid going through the laser beam. We have the filters followed by the detectors. And as you know, each detector is assigned to one fluorochrom. When talking about spectral flow cytometry, the main difference is in the way that light is being collected. So what are we looking at? Fluorochromes typically have more than one excitation wavelength and emit over a range of wavelengths. So when using the conventional idea of one laser, one detector, we are capturing only the peak emission. Okay, so it is like looking at the tip of the iceberg when there is actually much more beneath it. Let's take, for example, the fit emission spectrum and the detector bandwidth. When using a conventional flow cytometry, the detector only captures the peak emission of fits. So it makes us assume that fits is green. Anything that is outside of this detector is either being compensated or ignored. Now, let's start talking about spectral flow cytometry. It was first introduced to the flow community in this article mentioned here in 2013. And the article discussed the way that light is being collected across a detector array and not just through one detector. So if we look again at our FITS example, spectral flow cytometry collects the emission from a range of detectors. It's continuous bands of light where there are more than one detector for one color. You can see now that we can collect the entire emission, the full emission spectrum of fits from 490 nanometer wavelength to around 650. And we can also see that the color is not only green, but the colors of fits go from blue through green and yellow and orange. So let's dig deeper into full spectrum cytometry. We have the optical design. We have the lasers. Each laser has its own APD detector module that consists of several detectors. The detectors, the APDs, are highly sensitive, and the lasers, they're spatially separated. We have the full spectrum analysis for each fluorohorn. The entire emission spectrum is captured across the different modules to create the full spectral signature for each fluorohorn. We can see here that we have for the violet laser 16 detectors. We have 14 for the blue laser. We have 8 for the red laser. Last important thing to discuss here is the spectral unmixing. It's the algorithm that calculates the contribution of each known fluorohorn. It is actually the spectral unmixing algorithm. It replaces the conventional compensation that you are familiar with, and we will discuss this further later. So as mentioned before, Cytec is using highly sensitive APDs instead of the traditional PMTs for their Aurora and Northern Lights systems. The sensitivity of a detector is being measured by quantum efficiency. It is the ability of a detector to convert photons into electrons and finally into a signal. You can see on the graph here that APDs have much higher quantum efficiency than PMTs across a broader range of emission wavelengths. If we look at the efficiency of PMT, you can see that it drops down very early in around 500 nanometer wavelength. And if we take, for example, the Alexa 647, when using the traditional PMT, it has only 5% efficiency, meaning that out of 100 photons, only 5 of them will be converted into electrons. When using APDs, this number is very much close to 100%. This gives us much better sensitivity and resolution. We'll see it later. Another very important thing is that Cytec is using flat top beam lasers in their systems, meaning that the energy distribution is very homogeneous. Okay, it doesn't matter if you're using low, medium, or high flow rate. We will have the same energy, the same MFI, and the same resolution for the populations. For example, from negative to positive and dim populations. So in order to show you how the spectral signature is created, we will look at the 5-laser Aurora optic design. You can see the range of detectors in each detector module for each laser. The lower the excitation wavelength, we have more detectors in the detector module. So you can see for the UV and for the violet laser, we have 16 detectors. We have 14 for the blue, 10 for the yellow-green laser, and 8 for the red laser. Another more important thing to mention here that we have 2 side scatter detectors, one under the blue and second one under the violet, that will help us with small particles. We will show you some examples later. So let's take a closer look at how the spectral fingerprint is built. You can see here the emission signal in each detector. Here we see 16 detectors for the UV laser. And to understand this better, we'll put out just one of those channels. Looking at the top-down view of a regular count histogram, we can understand now that the red area in the middle is for the highest count, the most dense population area, where the blue has the least count. So in the next few slides, we will see how we are using the signature. First, we have to understand that spectral signature must be unique in order to distinguish between different dyes. We can see here just 4 examples of signatures for different dyes. We can see it again, the 5th example from before. We can see now that it not only emits in the blue detectors, it also emits in the violet detectors. We can see here the PE and the APC that has emission in the violet, in the blue, in the yellow-green detectors, and of course the APC also has emission in the red detectors. So first, and the important use for the signatures to evaluate dye compatibility. We have here 2 examples. If we take a look on the left, we have APC and Alexa 647. These 2 dyes are considered the same or interchangeable when using conventional flow cytometer. This is because the peak emission is almost the same. Here in spectral flow cytometry, we can see that there are quite few differences, meaning we can use them together, and Aviva will go over it later. On the right, we can see FITS and Alexa 488. We can see that these signatures are much more similar, and we can also see it in the overlay histogram. Another important use of the signature is to evaluate spread or spillover between 2 dyes. So here, for example, we have PSI5 at APC. If you attended our basic flow webinar last week, a question came up whether if you're using 2 dyes that are excited by different lasers, if you need to run compensation. Of course, the answer to this question was yes, and now we can also show you why. You can see here, in conventional flow cytometry, the PSI5 is being detected by the blue laser, and the APC is being detected by the red laser. But still, when looking at the entire spectrum, we can definitely see the spread or the spillover that PSI5, it has a lot of spillover into the APC. We can also see it here in the overlay graph, meaning that, of course, although they are being detected by different lasers, when you're using conventional flow cytometry, you need to run compensation between these 2 dyes. This, of course, doesn't interrupt us when using the automated unmixing algorithm on spectral flow cytometry. So I now hand over to Aviva.
So, hello everyone. Thank you, Nitzan. My name is Aviva, and I will talk about the scientific aspects of spectral flow cytometry. Now that we have talked about fluorochromes and how we can obtain unique signatures, let's talk about spectral unmixing and how it works for our system. Here, again, we are looking at the signatures to think about how the unmixing algorithm works. So how do we determine how much of each fluorophore is contributing to this signal? This is exactly what spectral unmixing is giving us. Similar to conventional flow, where single stain samples are used as compensation controls, in spectral unmixing, we are using reference controls, which are also single stained and unstained samples. We need these single stain controls in order to collect the signatures for each dye. Reference controls are absolutely critical to identify the individual spectral signatures. The spectral unmixing algorithm calculates the contribution of each non-fluorophore spectra to the total collected signal in each detector. So when we think about the algorithm, we can think of this as extracting each component until we have nothing left. So here we are looking at the compensation versus spectral unmixing. And it is quite clear that these different algorithms achieve the same thing. It's all about assigning photons to the correct fluorophore. In compensation, each fluorophore is detected in one detector, and the number of detectors is equal to the number of fluorophores. Whereas in spectral unmixing, each fluorophore is detected in all detectors. In conventional compensation, the algorithm mathematically subtracts the amount of spillovers into the primary detectors, whereas in unmixing, it determines which combination of reference controls best fits the signature of the multicolor sample. So here we have a very nice example of two dyes of Q dot 705 and the brilliant violet 711 in order to compare between the traditional compensation and the unmixing. These two dyes are heavily overlapping dyes, and as can be seen here in this lower part, we can see that the compensation doesn't work very well because the positive signals are not very well resolved from the negative signals, and the negative signal is not correctly clustered here, and here the double positive signal does not come up correctly. So when looking at unmixing, we can see that the positive signals are very well resolved and negative signals are correctly clustered. So the unmixing has the power to give us high quality data for these two dyes that cannot be combined together in conventional flow cytometry because the compensation does not have the power to resolve these two dyes and give you quality data. So here are some of the advantages of spectral unmixing. Overall higher resolution is achieved by using unmixing because more data points are collected for each measured particles. Since more photons of light are collected over the full spectrum, it gives tighter clustering of negative and positive populations. More colors with less lasers. Highly overlapping dyes can be analyzed together as long as the full spectrum signatures are unique. Any dye that can be excited by onboard lasers can be used. There is no need for optimized filter sets since signal is collected in all channels. It also allows us to extract the autofluorescence. The autofluorescence contribution to background noise can be minimized by extracting its signature from the sample. So let's take a look here on an example of the autofluorescence of three types of cells, of splenocytes, lung cells, and yeast cells. As can be seen here, the splenocytes have very low autofluorescence, but lung cells and yeast cells have higher autofluorescence, mainly in the violet laser channels. So please note that the patterns are quite unique. This is what enables us to treat the background signal as any other unique signature, meaning that the autofluorescence can be extracted using unmixing, same as with fluorescent dye. Here is a nice example of HeLa cells carrying an M-cherry reporter gene. So here we are looking at the unstained cells and the M-cherry signal. As you can see, both signatures are very similar. And look at the fluorescence signature in the yellow-green channels. There M-cherry signal is detected. You can see that the autofluorescence of the unstained cells is quite high in these channels also. So when we are looking on unstained sample, we can see high autofluorescence, and without autofluorescence extraction, the positive M-cherry signal cannot be easily resolved from the negative signal. But after autofluorescence extraction, we can clearly see our positive signal and how nicely it's resolved from the negative signal. And now we can gate on this positive signal very easily. So it is an extremely useful tool in case of dim populations, and also also in case of cells that have high autofluorescence. For an example, cells that come from inner organs, or cells from tumors, or tumor cell lines, or senescent cells. There are many types of cells that have higher autofluorescence, and it makes it more challenging to use dim, to resolve dim signals in case of high autofluorescence. So I think that this example here demonstrates very nicely that this technology is highly sensitive for dim populations. So now that we have talked about fluorochromes, their unique signatures, and unmixing, let's talk about the wide range of possibilities using Cytek instruments. So full spectrum analysis enables us to use highly overlapping dyes. This is something that San has already mentioned in her session, but now I would like to walk you through this superpower of spectral flow cytometry. Then looking at the emissions peaks of these two dyes, it is clear that they cannot be analyzed together in the same tube by classical flow cytometer. But when looking at the emission signatures of these two dyes, in the eyes of Aurora, very clear differences are seen. Let's overlap these two signatures in order to see the differences. So there are big differences in the ultraviolet, violet, and blue laser channels, but also the yellow, green, and red. In the yellow, green, and red channels, tiny differences can be seen. These differences are exploited in order to combine these dyes together in the same tube, and even to analyze co-expressed markers, as shown in this example here. Here is an example of PBMCs stained with CD8 APC and Alexa Fluor 647. Let's look at the cells that are gated on CD3, and we can see here how nicely the double positive signal is resolved. And this is something that we can only see in spectral flow cytometry, and this is impossible in classical flow cytometry. Another advantage in all Cytek instruments, let's look. In all Cytek instruments is the consistency of the signals between different instruments. This is both possible thanks to the fact that you don't need to adjust the voltages. The consistency of the signals between different instruments is of high importance for biotech companies that need standardized data between different instruments installed in different sites. Here you have an example for two different units which give identical output data for the fluorochromes listed here. Look at the signatures. They are exactly the same. This standardization is achieved thanks to the voltage that is controlled by QC beads rather than by the user. So small particle analysis is another application that we can benefit in Cytek instruments, thanks to the extremely powerful violet laser and highly sensitive violet side scatter detector. Here in this example here, we have a murine leukemia virus expressing a GFP fusion protein. The size of the virus is around 120 nanometers. And look here in the plot on the right side, how nicely the GFP positive signal is resolved from the negative one. Here is another example, even for smaller particles in a size of 81 nanometers. And these particles are non-fluorescent polystyrene beads, and it is very nicely seen here how only by the aid of side scatter the beads are resolved from the background. Okay, so this is also the place to mention exosomes that are of high interest today and can also be very nicely resolved and analyzed in Cytek instruments. High resolution of dim populations. So dim populations, this is always an issue when we build our antibody panels, because this is something that we need always to take into account. And then building these panels, we are usually choosing brighter fluorochromes for dim populations. So here in this example, we have a messenger RNA of a low expressed gene in lymphoma cell line. And when we are looking at the signature of the unstained cells and the positive Alexaflor 488 signal, we can see that they heavily overlap. They are almost identical. On the right, you can see that after autofluorescence extraction, a nice resolution is received, and we can gate on the positive signal and distinguish it from the negative one. So this autofluorescence extraction is really a superpower, and it is extremely beneficial when analyzing dim populations, as in this case here. In spectral flow, there are a wide range of possibilities. With spectral cytometry, the paradigm has changed. You don't have to think anymore of how many lasers you have. Instead, you can ask yourself how many markers you want to analyze in one tube. Here in this example, we have a panel of 10 violet dyes that have been used to stain PBMCs. These PBMCs are stained with these 10 violet dyes, and this sample was run on northern light that has only one laser, in this case violet laser, and 16 fluorescent channels. And look how nicely here the monocytes are differentiated, and how nicely the TCR gamma delta T cells come up, and all the other cell populations, the Tregs, how nicely they are resolved here and can be further analyzed. So you really don't need to worry about the optical configuration of the instrument, because any dye excited by the laser can be used. Here is another example for a panel of 21 colors, and it was run on an instrument with only 2 lasers. This is something that goes up and up because all the time new dyes are added. And of course, when you have more lasers, you can even go up until 40 dyes with a 5 laser instrument. But again, to achieve 21 colors in only 2 laser instruments, it is really amazing. So here are some additional advantages and possibilities. Volumetric counting. We have a volumetric counting that enables us to get the exact concentration of the cells and also the total cell number. Resolution is not impacted by flow rate, thanks to the flat top beam. One of the easiest things about the Aurora is that you don't usually need to adjust the voltages for the detectors. The Cytek assay settings and quality QC automatically set the gains for you. You have a plate loader option for 96 well plates that can be easily changed between plates and tubes in a matter of seconds. Also, washing and mixing between samples is possible. Automated QC and cleaning steps. Cytek Full Spectrum Viewer, it is a very, very useful tool. It shows you the level of similarity between different dyes, and it helps you to decide which dyes you can run in the same tube, together in the same tube. Also, IVD instruments. This is something that is already available since last week. So now an IVD Northern Light instrument is available for sale and can be implanted in laboratories. Cytek instruments are very well established today. Aurora's rapid acceptance in the market is a testament to Cytek's remarkable technology innovation. There are 3 years in the market and overall of 400 devices. So collaborations with companies like BioLegend and Thermo Fisher also are going on, and they assist to develop new dyes and antibodies that can be used in spectral flow cytometry. Also, Cytek have also established partnerships with software companies developing softwares for deep and advanced analysis like FCX Express from Genovo, Astrolab, Verity, and OMIC. All these are enabling cutting-edge analysis of deep flow data, and also what Nitzan showed in her second slide, I think it was, the high-dimensional analysis of flow data. This is also something that is enabled by all these softwares here, listed here. So now we will briefly look again into Aurora 5 laser system in order to understand its performance characteristics. So here we have 5 lasers that are spatially separated, and we have 5 detector modules. Each laser has its own detector module, and we have 2 side-scan detectors. One is under the blue laser, and the second one is under the violet laser. And this what enables us to analyze small particles. Here is a filter configuration for 5 laser Aurora. We have overall 64 detectors on the Aurora. So here, under the ultraviolet and violet laser, there are 16 channels, 16 detectors. Under the blue laser, there are 14 detectors. Yellow-green laser has 10 detectors, and the red laser has 8 detectors. What is also important to mention is that all these detectors have a bandwidth between 15 nanometers until 35 nanometers. And this enables us to get the full emission spectra from 380 nanometers until 850 nanometers. So this brings us towards the end of our presentation. I would like to show the new OMIP 69 that was published recently in Cytometry Part A. OMIP is an optimal multicolor immunofluorescence panel. It is a special review publication that reports on newly designed and optimized multicolor panels for flow. Cytec have recently published this review summarizing 40-color panel for deep immunophenotyping of major cell subsets in human peripheral blood. So let's look on the panel that they have designed. So you can see in this panel that there are brilliant violet 421 and super bright 432 are analyzed together in the same tube. FITS and brilliant blue 515 that have similar emission spectra or emission peaks are combined here, and also APC and Alexa 647 and many other ties that have highly overlapping emission peaks. Yeah, as you can see, there are still many channels available to be occupied by new fluorochromes that are hopefully under development today. So here is another way to look at the panel. Here are all the cell subtypes and the markers that are used to phenotype all the cell types. And here we can see is the analysis, is the gating strategy that was used. We will briefly go it through because it really bears a lot of data, but here, look at how nicely the monocytes, the non-classical and intermediate monocytes are resolved and seen here. Also, gamma delta T cells are very nicely resolved here. And many other cell types, regulatory T cells, B cells, dendritic cells. Also, we can see here the naive CD4 cells and also central memory CD4 cells here. So all these cell populations are very nicely shown in this paper, and in case you want to see it, you can find it through search in Google. So let's summarize the things that we have talked about today. Today we talked about the full spectrum analysis and how it helps to predict the dye compatibility. We looked at how full spectrum cytometry offers us a great flexibility to combine dyes that have very similar emission peaks. Autofluorescence extraction gives us much better resolution in case of dim populations, and also we talked about how resolution of small particles that is enabled thanks to the violet laser and the side scatter that is under the violet laser. So all these contribute to high quality and high parameter data. And don't forget, you don't need to think about how many lasers you have, but rather how many dyes you can combine in a given configuration. So thank you for listening. And you can contact us at our emails listed here. If you have any questions or you need any further information, you are welcome. We are always happy to assist.
Thank you.
Thank you.